Breeding plants and animals for food. Plants and animals are bred to improve characteristics to help support sustainable food production. Breeders develop crops and animals with higher food yields, higher nutritional values, pest and disease resistance, and the ability to thrive in particular environmental conditions. The characteristics selected by breeders would include increase in yield, resistance to pest, and resistance to disease. Genetic variation plays a very important role in this. There are two types of variation: discrete variation, which are features usually controlled by a single gene that do not cover a range of values, or continuous characters which show continuous variation, which are usually controlled by more than one gene, covering a range of values. So once again, a characteristic that shows discrete variation controlled by the alleles of a single gene, which may be dominant or recessive. However, characteristics which show continuous variation are controlled by the alleles of more than one gene. Single gene inheritance can be shown by a monohybrid cross, where true breeding original parents for the dominant and recessive phenotypes can be bred together to produce ultimately an F2 generation, which shows a ratio of 3:1 dominant to recessive phenotype. Any characteristic showing discrete variation largely unaffected by environmental factors, but a characteristic showing continuous variation can be influenced by the environment. In selecting and breeding, a breeder will select plants or animals with a desired characteristic and use them as parents to create the next generation. And the aim is to produce offspring with a combination of the alleles of the genes for the desired traits from each parent. Inbreeding involves the fusion of two gametes from close relatives. However, outbreeding involves the fusion of two gametes from unrelated members of the same species. In inbreeding, selected related plants or animals are bred for several generations until the population breeds true to the desired type due to the elimination of heterozygotes. This process is repeated over and over again until you reach offspring showing the desired characteristics. Inbreeding ensures that each generation of a selectively bred organism receives the alleles for the desired characteristic. Inbreeding to produce a breed of animal, true breeding for desired characteristics, is carried out as in the diagram we've previously seen. This has the effect of concentrating the alleles for the selected traits into one line of breed. The effects of inbreeding include loss of heterozygosity. Continuous inbreeding leads to a loss of heterozygosity and the development of homozygosity. With each generation of offspring bred from inbreeding, there is a loss of heterozygosity. However, homozygous dominant and homozygous recessive alleles remain in the offspring. A result of inbreeding can be an increase in the frequency of individuals who are homozygous for recessive deleterious alleles. These individuals will do less well at surviving to reproduce. This results in inbreeding depression. This can be illustrated in this example where maize, a natural outbreeder, is forced to undergo inbreeding over a number of generations. To reduce the effects of inbreeding depression, new alleles are introduced into the plant or animal line by crossbreeding it with a strain possessing a different but desired genotype. An F1 hybrid is an individual resulting from a cross between two genetically dissimilar parents of the same species. Breeders often cross members of a species that have two different desired characteristics to produce some F1 hybrids that have both. In plants, F1 hybrids produced by the crossing of two different inbred lines create a relatively uniform heterozygous crop. F1 hybrids often have increased vigor and yield. Plants with increased vigor may have increased disease resistance or increased growth rate. And this can be illustrated in this example of hybrid vigor in maize plants. In animals, individuals from different breeds may produce a new crossbreed population with improved characteristics. The two parent breeds can be maintained to produce more crossbred animals showing the improved characteristic. In inbreeding animals and plants, F1 hybrids are not usually bred together as the F2 produced shows too much genetic variation. As a result of genome sequencing, organisms with desirable genes can be identified and then used in breeding programs. Breeding programs can involve crop plants that have been genetically modified using recombinant DNA technology. Single genes for desirable characteristics can be inserted into the genomes of crop plants, creating genetically modified plants with improved characteristics. Recombinant DNA technology in plant breeding includes insertion of Bt toxin gene into plants for pest resistance, glyphosate resistance gene inserted for herbicide tolerance.